we get experimental science. We're curious, non-judgmental.
There is a species of ant that's being studied in like the molecular genetics labs where a worker can become a queen. It's very rare for that to happen. Most of the time you're either born a queen or you're born a worker, and that's that. But this ant allows there to be a transition. And when the worker becomes a queen, her lifespan multiplies by 6. And that's all due to genetics. What actually ends up happening is that the genes in the genome are there in the worker and the queen. They're just off in the worker, and they're on in the queen. So when that ant transitions from worker to queen, those genes that were off turn on, and it extends the lifespan. And so it turns out that that gene is present in humans, but that it's very tightly regulated. So imagine if you're able to turn on that gene in a human. It's already there, it's just usually in an off state. That will end up maybe extending lifespan. The catch, though, is if you turn it on everywhere, it turns out it gives autism-like symptoms. So you not only need to know how much to turn it on by, but where, right? So if you can turn it on the right cells, then you might be all right. How do they decide to be who's the next queen? So they fight with their antennae. Here are the ant species. You can see in this video, there are the eggs and the larvae. Here are the pupal cases, so that's like the chrysalis, the cocoon. The yellow individuals are newly hatched, and the brown individuals are mature adults. So that's just a little context for you of how to read this particular video and just see what the animals look like and what that means. So here's the species, and you can see there's a lot of interaction going here. See how big their eyes are? So they're a visual hunter. They are the length, the first 2 knuckles of your pinky is a rough estimate, and I know everyone's pinky is different sizes, but like an adult hand's pinky, they're quite large. They're thick, thin. They're a very ancient ant species. They're more closely related to wasps than anything else. They are Harbagnathos saltator. They are commonly called the Indian jumping ant. Yeah, they're quite large. You see their mandibles, those jaws? Those they don't bite with them. They lift. They can lift each other and catch prey. I will actually show off a hunting video if y'all are interested. So that's what the ants look like. There's also, you see crickets in here. That's what they eat. They're also, right here, you can see this individual is tearing apart a cocoon. The cocoons are reused. They're utilized to make new cocoons. They can spin a little bit of a silk web, but they need wood shavings and other things to make a solid cocoon-like structure. And so to do that, they actually reuse some of their cocoons. And then here you can see they have a stinger. So they have a stinger that injects paralytic venom into their prey. It's because they don't eat anything that's already died. They only will eat living individuals. So they paralyze, the venom paralyzes the crickets. They can't function anymore. And the cricket will die paralyzed. Once the cricket dies, they'll get rid of it and put it into a trash pile if they haven't finished eating it. Here, this is outside the nest. These are older animals. Those are going for foraging. They're going hunting. They're going out exploring here, playing a little game of peekaboo. There, just to give you all the vision, that's the mandibles right there. Look at how long those mandibles and jaws are right there. And again, that's not for biting. They hold on to one another and lift up, and that's how they do these interesting wilding behaviors. Again, her eyes are huge. So there's also the stinger. That's where the venom is. Actually, I've been stung a fair bit by these. It burns a little bit when you get stung, but it's not really painful, but it does welt up. So it's almost like a bee sting. Interestingly enough, I haven't heard of anyone having an allergy to it, but I'm sure there are some folks that have allergies to this particular kind of ant sting. So just as a possibility to watch out for, you know. Here, this winged one is a very special one to actually see here. The winged individual here is the true queen. The true queens are very rare. Because any worker can become a queen, you're not likely to get these, but this one is born as an actual queen in the colony. That's why she has wings to do her mating flight. There again is the colony. Here, they're taking care of the embryo. That is the brood or the larva. These two are going to be fighting for the right to become queen. They fight with their antennae for over the course of 7 to 10 days. Every day, fewer and fewer are fighting until at the end, there's only one winner. And that one winner is the one that becomes the new queen. So watch them. Right, there you go. That back and forth behavior right there is the fight. The only time you will see that is if something has happened to a queen and they need to generate a new queen of the colony. That butt wiggle is also an aggressive form of posture. So actually, if you open up the nest and look at the ants, they will look at you and they will shake their butt at you and be like, get out of here. I don't want you here. You're not supposed to be here. Because they're so visually attuned that they can actually see us. That's what the fight looks like. And again, it's over the course of 7 to 10 days. Fewer and fewer fight each day until finally there's a victor. The one that wins, their lifespan multiplies by 6. Their brain shrinks. They lose about between 30 and 15% of their brain mass, and their ovaries get activated. Yeah. Other ants, like carpenter ants, ones that are underground, primarily have much smaller eyes. They see you as a shape and not in a crisp resolution. And she gets wings. No. She doesn't get wings. She does not get wings. Only the true queens that hatch will have wings, and those will have a mating flight. Those don't need to fight because they are a true queen. This transition from worker to queen requires a flight, and that will not yield to any kind of flight. They won't grow new wings. So when they become a queen from the fighting, that's that. They don't get to grow. They don't disseminate their genetic information. They become a queen in the colony that they're already in. They don't move elsewhere. How do you measure this? So Balmer, there's a couple of ways to measure this behavior. A lot of it is through AI, machine learning, where you feed it into a computer software that will track movement of the animal, including but not limited to the antennae. And then you can measure antennal interaction between different individuals. And that's, I think, a really cool part of doing it. An original way of doing it was by eye. So you watch them and you saw how often they interact. And so it's better with the new software because it's unbiased, which is really neat. Flight or flight. It kind of is, Cliff, yeah, but not in the same context, of course, as the fight or flight of adrenaline. So we don't know what they're fed. There is a hypothesis that, and not just in this species, in ants, we do not know what the cue is to become a certain caste. Unlike in bees and honeybees, if you're fed royal jelly, that royal jelly will make that larva become a queen. However, in ants, we do not know what that signal is. Again, optic, we think it's through feeding, because you have a nurse feeding larvae, and that seems to kind of start delineating what they end up becoming. But you can't not have the larva be fed by a nurse, right? So that would be one control, is if you can remove that feeding behavior, do they become a base caste or something else? You can't stop that because they have to be fed by a worker. You can't just give them food sources and have them eat on their own. So again, there's this interaction between the nurse and the brood, and the hypothesis is that there's epigenetic factors that are in the saliva of the nurses, and that dictates what the individual is going to become. Hasn't been clearly enough shown to be like that's really convincing, but that's the hypothesis we're operating under. New queen is sterile, so when any of these, so they're not sterile, but they're unmated. Any one of these workers can mate. They just don't unless they become a queen. If there are no males to mate with, the ants can lay unfertilized embryos, will become male. And so what they can do is they can force males to be produced, mate with them, and then they can produce females. So the genetics work, it's not XX and it's not XY. X nothing is a male, XX is female. And that's just a unique Hymenopteron genetic trick. There are parasitic wasps that function the same way, but then if you go to fruit flies are XXXY. So it's all over in the insect world. I wonder if they are taking jobs and evolving them into them, so to say. Evolving, I'm sure at some rate, optic, but it's questionable as to how quickly, of course, that works. They most likely do, yeah. And Dramic, these are the jumping ones. So when these individuals, after winning their fight, if they become the new queen, they become, well, I guess 6 times lifespan multiplied. Short-term evolving, short-term is very subjective. I guess optic, that's the catch on that front, right? So I guess I've heard, it just depends on what scale you're working on. Like if you're talking short-term in a yeast lab, you can have yeast evolve in a week, right? If you're selecting for a particular trait in a week, you'll have a whole line that's been created for that. Probably selection is the cleaner, easier word. You try giving them cheated wins, like gluing. So Dramig, there is a cheat you can do with these. If you isolate a worker, if you take any one of these workers and put them by themselves, they will become a queen because there's no signal for them to say no. They have not been beaten. If you, again, have them by themselves. So Dramig, you cannot look at a colony and tell who's going to be the winner. You cannot tell. Otherwise, Dramig, right, then if you can predict the winner, you can put a handicap on them and see what happens. But it's very hard to predict. Right now, what researchers are trying to do is use AI, machine learning, behavioral tracking to figure out if there's a pattern. Like, if you let them start fighting, could you predict on day 5 who's going to be the winner at day 10 based on that interaction? But we don't know that yet. Handicap all but one. So Dramig, some don't fight. Most of them do, but some just don't care. It has to do in part with age. There's also, to make it even more confusing, what you can do, if you take a queen of this species and you put them into an established colony that already has a queen, what's going to happen is they're going to hold on to her. They're going to police her. They hold on to her with her mandibles and until her ovaries revert and shrink back down. So you can have reverted queens, meaning that they were a worker, they became a queen, they reverted back into a worker, and where it gets really wild is if they have lived longer than their expected lifespan, they will die, right? So Dramig, think about that. If your lifespan, let's say, is 2 years, you become a queen, your lifespan is now 6 years, and you've lived 2 years, 1 month, and then you get reverted back to a worker, you die. It's pretty wild. These worker queens also live for as long as the real ones. Yes, it is a member of the RNA processing complex, where its job is to chop up RNA molecules and to decrease them, and so that they never protein gets made for them. It's a second form of it. So form number 1 is expressed in both worker and queen, but form number 2 is only expressed in queen. And this molecule has particular targets that will nerf those kinds of RNA molecules, and that's what we think allows for the extension of the lifespan. I do not, again, remember the name of what that molecule is, but that's how it ends up working. Yeah, and then so as soon as they go back to workers, that gene actually, Gwen, gets turned off again, and so if they've lived past a certain lifespan, they're gone. How do you understand ants like this? So Balmer, I'm an ant geneticist. So I did 10 years in a fruit fly lab and 4 years in an ant genetics lab. And actually these species that we were looking at, these I actually was part of my research. So I studied 3 different species of ants: the Indian jumping ant, the carpenter ant, and leafcutter ants. So as Mike's just dropped, I have my PhD in molecular and systems biology. And, genetics, neuroscience, behavior are kind of my wheelhouse. So how can we understand ants? What do they do, and why do they do what they do? Balmer, there are studies that we can do to modify their genetics. For example, there was a study that I was working on where we were trying to analyze what genes were best involved in leafcutter ant behavior. So what makes a soldier behave like a soldier, what makes a nurse behave like a nurse, and what makes a leafcutter behave like a leafcutter. What you can do is you can measure the genetic activity in their brains. That's what we did. We took out their brains and sequenced them and found the different RNA molecules that were being differentially expressed. So the DNA is the template that you make RNA from. There's different RNAs in different castes of ants. So soldiers had one RNA that was really, really, really high. There's a lot of that gene turned on. It was near off in a nurse, and it was an intermediate in the cutter. And what you can do, Balmer, is you can inject molecules into the brains of the animals to turn down a gene or to turn it up. And actually what we did was we turned down the gene in the soldier and we got it to behave like a nurse. We also then turned up the gene in the nurse and made it behave like a soldier. So that's kind of the way we can study. So you can do these injections into their brains. You can also try to make mutants, so you can genetically modify the animal. But it's very hard to do in ants because it's impossible to mate them in a lab setting. It did work, Balmer. Yes, it did work. It's all due to a neuropeptide in the brain.